ஸ்லோவா இருக்கு. ஆமா, நான் இங்க எழுதினாலே ஸ்லோவா தான் இருக்கு. தெரியல இல்ல, சரிங்க, இங்க வருதா பாருங்க ஒரு நிமிஷம். தெரியுதுல்ல. தெரியுதா சார்? Stop பண்ணு. Okay, still listen. Ready? So I didn't come for last class last week. Biology exam over. So which chapter? Life process. Okay, now they started control and coordination. Okay, so in life process, how you did? Did well? Extremely well? Everything? Hmm. Okay. Fine. So anyway, you people missed both all of the genetics classes, right? Genetics. That's what. So 2 weeks, no genetics class. Both of you. So we were as much slow as possible. Just a minute. ஆமா சார், டியூஷன் வந்துட்டேன். 8:30க்கு முடியும். நீங்க வரீங்களா? கண்டிப்பா வாங்க சார், கண்டிப்பா வாங்க. ம், கண்டிப்பா. ம், ஓகே. நீங்க ஒரு 8 o'clock இங்க 8 நம்ம குருநானக் காலேஜ் கிட்ட வந்துருங்க. 8 o'clock 8:30க்கு அப்படி வந்துருங்க. நாங்க வந்து உங்களை பிக்கப் பண்ணிடுறேன். ஓகே ஓகே சார், தேங்க்யூ. கண்டிப்பா வாங்க. Bye. Okay, fine. So coming to this, actually, genetics, heredity, many things we have discussed actually in the class. Like some simple introduction I'll give you, like checklist, what you have to know. So that I will tell you. Okay, so before getting into today's class, so what's genetics or heredity? The chapter is heredity. Okay, chapter is heredity. See, for you only, please listen. Okay, yeah. See, what's evolution? Evolution is the study of origin of Earth, first life form, and diverse from that first form, how the diverse live forms were formed. This is called what? Evolution. Evolution. Okay, evolution. This evolution has three components. Rohit, what are the three components? Heredity, variation, natural selection. Okay, what are the three components of evolution? Heredity, variation, natural selection. What's heredity? How characters are transmitted from parents to progeny. How human beings produce another human being. That human being, another human being. So this is heredity. Okay, fine. And then variation is when the organism produces a progeny. In what way the progeny vary characters from the parent? That's variation. Heredity, variation. And one simple thing called accumulation of variation results in speciation. Accumulation of variation results in speciation. Okay, so because of variation, new species forms. Accumulation of variation, little, then all little things cumulatively contributes for formation of new species, new origin of new species. Okay, now you have old species and you have new species. Because of variation, new species is formed. And natural selection means whether the natural selection gives survival advantage of old or new, or both. It supports both, either old or new, or both, or will not support both of the things. What is going to decide? Nature. Nature only select this organism is fit for the environment, let it live. If the environment does not support, the organism will extinct. Otherwise, it will exist. If it supports, the organism adapt to the environment, it can survive. So, all these things are this particular chapter. Okay, you should know what is evolution and what is heredity, variation, natural selection. General basic thing you should know. And apart from that, heredity, say characters are transmitted from parent to progeny. For that, they follow rules. Okay, for that, they follow what? Rules. Not just like that, they transmit character. Characters are not transmitted as such. They follow certain rules to transfer the characters. Okay, fine. That rules of heredity, how we understood. Okay, based on Mendelian experiments. Mendelian experiments. Okay, who is Graham Mendel? Who is Graham Mendel? The father of genetics. He is an Austrian monk. Okay, he performed various experiments using a pea plant. Okay, what plant? Pea plant. He used various experiments. Okay, he performed more than 25,000 experiments he performed. How many experiments? 25,000 experiments he performed. For 7 years he performed. What is the scientific name of this pea plant? Hari, what is the scientific name of the pea plant? Pisum sativum. Okay, Pisum sativum. This is the scientific name. Garden pea plant is the common name. Tamil என்ன சொல்லுவாங்க? பட்டாணி செடின்னு சொல்லுவாங்க. Can you understand? In the plant la he was doing. And he selected 7 characters. Okay, in this pea plant, he selected 7 characters. What are the 7 characters? Seed shape. And then Rohit, tell me, what are the 7 characters? Seed, color, and then pod shape, and then pod color, and then flower position, and then flower color. Okay, and then stem height. What are the 7 characters selected by Grover Mendel? Seed shape, seed color, pod shape, pod color, flower position, flowers color, and then stem height. See, interesting part of the plant is it showed two contrasting characters. Seed shape, either it looked round or it looked wrinkled. Round means proper round shape. Wrinkled means, can you say this is wrinkled? And then seed color, either it looked yellow or green. Okay. And then pod shape. What is pod? The one which protects the seeds. Okay. Yeah. Here you have the seeds, that covering. That is called pod shape. See, this is full. Okay. And some will have that such kind of appearance. The pod is like this. Okay. This is called what? Constricted. What are the two variation in pod shape? Either full or constricted. It looks plain or it looks like this. Okay, constricted. And then pod color, green or yellow. Can you understand? Pod color, green or yellow. Flower position. There are two. What are the two? Axial, terminal. What is axial? What is terminal? See, this is the stem of the plant. Okay, this is the stem of the plant. If the flowering occurs along all the axis, it is called what? Axial. Okay, are you listening? Terminal in the sense, if this is the stem, only in the terminal end you see the flower. Can you see? All the flowers are in the terminal end. No growth of the stem after that. So, two variation, axial, terminal. And flower color, majority it is violet or purple. And then another contrasting thing is white. Stem height, tall or short. Either tall plant or short plant. Can you see? So what are the 7 characters? Seed shape, seed color, pod shape, pod color, flower position. And then stem height, 7 characters. See, the first listed characters are what? Dominant character. See, in every organism, in Mendelian genetics, organisms will have 2 contrasting characters. One character is dominant, another character is recessive. One is dominant, another one is recessive. Okay, here, what is dominant? Majority of people in the population, the characters in majority, it is present. Another one is rarely it is present. Always something is present, rarely something is present. That is dominant, recessive. In simple, okay, widely seen, rarely seen. That's dominant and recessive. In simple layman term. Can you understand? So, seed shape, what is dominant? All the first listed thing are dominant one. Can you understand? Fine. So, he selected all these characters and performed experiments. Okay. So, to understand these experiments, you should know about many terms in genetics. What are the terms? Genes, alleles, okay, genes, alleles, and then homozygous, heterozygous, okay, dominant, recessiveness, genotype, phenotype, okay, genotype, phenotype, mono, hybrid, cross, di, hybrid, cross, back, cross, test, cross, reciprocal, cross. Okay, Punnett square, filial generation. Okay, can you understand? So like that, many terms. If you understand the terms only, you can understand what genetics is. Okay, fine. And then, you know, self pollination, cross pollination, pure line, etc. Okay, emasculation. Have you know? Do you know about all these terms? Bagging. Okay, கேள்விப்பட்டிருக்கீங்களா இந்த terms எல்லாம்? Anyway, just listen. Okay, quickly we'll finish these terms. So, genes are what? Genes. Factors determine character. நம்ம கிட்ட இருக்கிற எல்லா கேரக்டரையும் டிட்டர்மைன் பண்ற செக்மெண்ட் ஆஃப் DNA is called what? Gene. Fine. Alleles, alternate forms of gene. For example, one gene determines blood group A. Okay, another gene determines blood group B. So A gene is a gene, B gene is the allelic form of a gene. Can you understand? Homozygous in the sense, for one character in a parent in a diploid organism, two same alleles. Okay, two same alleles. For every character, how many genes we have? For every character, how many genes we have? How many alleles we have? Two. One from Appa, one from Amma. For every character, we have two genes, two alleles. One from Appa, one from Amma. For every character. If both are same type, it is called what? Homozygous condition. Homozygous. T represents actually here, since we use Mendelian concept, so T means tall plant. Okay. And heterozygous in the sense, one tall gene, one dominant gene, one recessive gene, one small gene. Can you understand? One big gene, one small gene. So this is called what? Two different alleles. It's called what? Heterozygous condition. Dominance in the sense, widely seen. In simple, widely seen. Appears in Mendelian genetics, appears in F1 progeny. Widely seen, appears in F1 progeny. What is F1 progeny? When two parents are crossed, the first generation is called F1 progeny, filial one generation, first progeny. Okay, in the first generation, some characteristic that is dominant gene. Recessive express are shown express only in absence of dominant gene. Dominant gene illenna modha recessive gene express agum. Okay, big gene, not strong gene, big gene, or the recessive gene, or recessiveness. That character is recessiveness. Genotype. What is genotype? Genetic composition. Genotype means what? Genetic composition. நம்ம கிட்ட இருக்கிற ஜீனோட composition is genotype. Phenotype அப்படின்னு சொன்னா visible character. For example, black hair, brown hair. நம்மளால பார்த்தா கண்டுபிடிக்க முடியுமா? Can you be able to identify black hair, brown hair? பார்த்தாலே தெரியும். That is visible character. Can you understand? That is phenotype. In Mendelian genetics, round shape, wrinkled shape. Can you see and observe? Can you see and observe? Yes. So that visible characters are called what? Phenotype. That is determined by genotype, which you cannot see. Okay, genotype, that genetic composition. Monohybrid cross, for one character, to determine one character, a cross is done. Inheritance, to study the inheritance pattern of one character, one pair of character. Cross of one pair of character. Dihybrid cross, or YY, or YY. So two pairs of characters. That is dihybrid cross. Hybrid, you are going to make a hybrid with two characters. Okay, fine. That's dihybrid cross. Back cross in the sense, Rohit, did we see back cross? What is back cross? See, two parents are crossed. Okay, two parents are crossed. First generation is come. Okay, one progeny comes. F1 progeny. Progeny means what? Offspring. F1 progeny into any parent. That is called what? Back cross. Progeny with parent is called what? Back cross. Can you understand? Progeny with parent is back cross. Test cross means you select unknown, means unknown plant into known recessive. Say you select a plant which is recessive, like always it produces only dwarfness. Select that plant and cross it with unknown. I'm giving you a plant to determine what is it. That is test cross. Okay, fine. What is reciprocal cross? Rohit, did we see? What is reciprocal cross? For example, see first male into female. For example, male is tall, female is short. Okay, this is the symbol for male and female. You know that, right? Now, reciprocal cross in the sense, now male is dwarf and female is tall. Vice versa. This is called what? Reciprocal cross. What is Punnett square? A scientist called Reginald Punnett developed a simple square method to predict phenotypic outcome. Tabulation. Okay, we basically work out genes and its composition. Prediction of genetic composition is called what? Punnett square. Introduced by Reginald Punnett. Name of the scientist, it is called Punnett square. Filial generation, first generation. Filial one means progeny, basically. Self-pollination, same plant, same flower. Okay. Cross-pollination, different plant, different flower. Pure line, how many generation you cross it, same character only produced. For example, if it has homozygous condition, that is called what? Or it can be this one. This is called what? Pure line. Okay. Emasculation is removal of anther, male part. Removal of anther in a flower. That is emasculation. Bagging is protection of stigma. Can you understand? Or female flower. That is what? Bagging, covering the flower with a bag or whatever. Can you understand? So these are all the terms you should know. Okay. Now, Grover Mendel studied how these characters inherited. Okay. So, first experiment is actually called what? Mono hybrid cross. What is it? Mono hybrid cross. So I'll show you how to work out, and same thing you also should try. Mono hybrid cross. Okay. See how you can work out. First you should write P generation. P generation means what? Parent generation. Okay. You are crossing it for one particular tribe. Select the height of the plant. Okay. Into. Okay. How do you describe this? This is genotype. How do you describe? Homos. For example, this is a male plant. You can describe this as homozygous dominant for tall. Okay, this is homozygous recessive DOF. DOF are what? Short. This is how you have to describe this. And then how the gametes will form. There are two ways by which two type of gametes can be formed. See, T, T, and then T, T. Okay, gamete formation. Here you have to write gametes. They are the gametes, right? Now, F1 progeny forms. How F1 progeny? The gametes will fuse and the F1 progeny forms. This one with this one. This one with this one. Now it is okay. How do you describe this character? TT, heterozygous. Heterozygous tall. Since if one T, capital T is there, definitely what is expressed? Only the dominant tall character is expressed. Even though it has small t, it will not express. Can you understand? Now Mendel actually wondered why when two plants like this pure line, they are two pure line are crossed, why only one character is expressed? Okay, and then selfing of F1. Here what occurs? Selfing of F1. What is the step now? Selfing of F1. Can you all able to follow? Fine. So how the gametes will form here? How the gametes will form? One capital T, one small t. Okay, and then one capital T, one small t. Can you understand? Now, for this selfing, you are going to do what? Punnett square. What is Punnett square? To predict the F2 progeny outcome, you do what? Punnett square. Okay, Punnett square. How it looks like the Punnett square? Okay, and these are all the gametes, right? One male parent and then one. So, for example, this is male parent and this is female parent. Okay, put the genotypes here. Okay, and here the alleles. Can you understand till this? And now, how do you work out in the Punnett square? This T and this T. Yes, and now this T and this T. And now this T actually, but always dominant should come first. Okay, and now what? Small t, small t. Is it clear? This is how you have to work out the Punnett square. Now, what next you have to do is write the genotype. Genotype. How many are there genotypes? So this is the first genotype. Yes or no? One genotype is there. How many are there? One. And this is the second genotype. How many are there? Two. Two. And this is the third genotype. How do you write it? How many? 1. So this is called what? Genotypic ratio. For monohybrid cross, what is the genotypic ratio? 1:2:1. What is the 1? How do you describe this? How to describe this? Homozygous dominant tall. How do you describe this? Heterozygous tall. How do you describe the third one? Homozygous recessive dwarf. Short. Can you understand? Like that you have to describe. And now write the phenotype based on that. What are the phenotype? See, how many tall plants were there? 1, 2, 3. This one also tall, this one also tall, this one also tall. Tall, one phenotype. What is the other phenotype? Dwarf. 2 phenotypes. Okay. How many were tall? 3%. 1, 2, 3. All were tall. And one is dwarf. This is called what? Phenotypic ratio. Can you understand? See, the significance of this monohybrid cross. What Graham Mendel concluded with this? Hari, what is Mendel's conclusion? He derived 2 laws, okay, based on monohybrid. One is law of dominance. Okay, what it says? Characters are determined by factors. Okay, what is the first postulate of law of dominance? Characters, every organism, characters are determined by factors. Factors exist in pairs. Factors exist in pairs. And then trait or character expressed in F1 progeny is dominant. Okay, character expressed in F1 progeny is dominant, means one is dominant. Character expressed in F2 progeny is recessive. The character, there are 2 characters. Okay, factors exist in pair. What expressed in F1 is dominant. The one which is expressed in F2 is recessive. Okay, what is expressed in F1 here? Tall character. That is dominant. Dwarfness also expressed, shown. That is in F2. Okay, that is recessive. This is law of dominance. And now, second law. Law of Kaushik. What is the second law? Oh, are you listening now? Second is law of segregation. Segregation. What is the spelling for segregation? SEG. Segregation. Correct? Law of segregation. Look at that. What it says. Generally, what we think actually. If a character is dominant, say if a character is dominant, that only expressed in large amount, means inherited in large amount. Dominant character inherited in large amount. Recessive character rarely it is inherited. We think so, but that is not correct. That is not the proper rule of genetics. Look at that. Here, say this is F1. Yes or no? This is F1. Here, how many tall genes and how many dwarf genes are present? 2 capital genes are present, 2 dwarf genes are present. Means in what ratio it is present? 1:1. And look at that here. How many dwarf gene and how many capital T? How many capital T is present? How many small t is present? 3 are. There are totally 8, right? 1, 2, 3, 4, 5, 6, 7, 8. 4 capital T, right? 4 small t. Then what is the ratio? 1:1. 1:1. Equal. Can you understand? So, no matter it is a dominant gene or a recessive gene. Okay. How it is, say, by what process gametes are formed here? Gametes formed by meiosis. During meiosis, from a parent organism, both of the gametes segregate and equally inherited to the next generation. It may be a dominant gene or it may be a recessive gene. They undergo meiosis equally and then they are inherited equally to the next generation. So, gamete formation and inheritance does not show any variation in dominant gene or recessive gene. Equally, they follow the same rules. That law is called what? Law of segregation. Okay, law of segregation. How do you write law of simplify? Law of segregation. Okay, it may be a dominant gene or dominant allele, you can say dominant allele or recessive allele. Okay, they equally segregated during meiosis and equally inherited to the next generation. Simple. That's the simple thing about law of segregation. It may be a dominant gene or equal gene. They are segregated during meiosis, and every gamete receive one copy. Each gamete receive one copy of one gene, and they equally segregated, equally inherited. That is called what? Law of segregation. Can you understand? These are all the 2 laws made by Graham Mendel, means suggested by Graham Mendel, and they are called first law of genetics and second law of genetics. Can you understand? I think this is what we have discussed in the previous class. Okay. Now, of course, it is due for all of you. That's for inheritance, to study the inheritance pattern of one trait, one pair of contrasting trait. Now we are going to discuss about what? 2 pair of contrasting trait. That experiment is called what? Dihybrid cross. Okay, that is called what? Dihybrid cross. I'll give you an assignment. Maybe tomorrow's class, you have to work out monohybrid cross and dihybrid cross and show me in the class. Okay, monohybrid cross and dihybrid cross. Okay, look at me and then how I'm just doing the dihybrid cross experiment. Okay, and this is based on what? The characters which is present here. So listen, in your textbook. Okay. Here, 2 characters are there, right? Okay. There are 2 characters, right? Look at that. What are the 2 characters, 2 pair of characters taken here? Shape, seed shape is round. Seed color is green. And seed shape, another alternative form. For round, what is the contrasting character? Wrinkled. And another one is actually called yellow. Can you understand? Round green, wrinkled yellow. In any way, you can shuffle it and take it. Can you understand? So you're going to work out this and you have to bring me the notebook tomorrow. Can you all be able to do that for both monohybrid and hybrid cross? See, look at that here we saw, right? F1 3:1 ratio for monohybrid cross and for dihybrid cross. So both you have to work out and show me. Okay. Now listen, quickly I'll do it. Please follow. And then tomorrow you try at home. Look at that, how I'm just doing it. P generation. The characters are, I'm just going to take 2. Okay. And then what are the recessive character? Little varying from your anyway, if you take, it should come. See, in your book it is one dominant, one recessive, one dominant, one recessive. But I'm going to take it both dominant, homozygous dominant characters and homozygous recessive character. In either way you want, you can do it. Are you all listening, both of you? All of you? Fine. Now, how the gametes will form here? Okay, how the gametes will form? Okay, this one, this one, this one, this one. Okay, similarly like this, how the gametes will form? Or Y, and then this or this Y, like that gametes forms. Okay, and then F1 progeny forms. How the F1 progeny forms? Look at it, or Y, Y. You know how I have put this or here, this or here, this Y here, this Y here. Can you understand? And then here again this or, and this Y and this Y. How the characters? They are heterozygous for round. What will be the shape? No, wrinkle will not express since one dominant gene is there. Round and yellow. See, listen. Please keep that in mind. Or round, dominant, or wrinkled. Y, yellow, dominant. Y, green, recessive. Can you understand? Never, this is the formula which actually you should know. That's why first 7 characters we used. Okay, there you should know what is dominant, what is recessive. Okay, all were what here? Round and yellow. Heterozygous for round and yellow. Okay, fine. Now, selfing of F1. Okay, what is the second step? Selfing of F1. See, this heterozygous should be selfed. Selfing of F1. And before that, how gametes will form here? What are the composition of gametes? For example, taking this, how gametes will form? Or. Can you understand this or with, and then this or, this or. Okay, this one twice I'm just writing, and then capital Y, small y, capital Y, small y. So you should know how the gametes are formed. Are there any other option? Either this R can go with this Y or with this Y. And this R can go with this Y or this Y. That's what I have written here. This R with this Y, this R with this Y. Capital Y, small y. One dominant allele, one small allele. That's what I have given here. So 4 gametes. 4 type of gametes can be formed. Okay. If here in this parent 4 type of gametes, and definitely same in this parent also, because both are heterozygous, same genetic composition only it has. Now, to predict F2 progeny, to predict F2 progeny, what we should do? Punnett square. Okay, Punnett square. See, listen how I'm just doing this. I'm making 16 squares. Okay, and then okay, and then okay, and then is it clear how many small squares are there? 16. Oh, yeah. Sure. In hurry burry, I clicked. Are you all following? Can you all able to do it independently at home tomorrow? Let me try. See, can you all able to see? I have to share. See, can you able to see? Now, see four gametes combination we wrote. Here, the first one I'm going to draw. First one. What was the second one? Capital O, small y. Right. Third one, small r, capital Y. Fourth one, same thing you write it here also. Or Y. Capital O, small y. Okay. And then small r, capital Y. Small r, small y. Now we are going to put the genetic composition. First these two. Okay, look at that. Tell me capital R, capital R, capital Y, capital Y. And here capital R, this one, capital R, capital Y, small y. And third one is actually capital R, small r, capital Y, capital Y. Fourth one is actually capital R, small r, capital Y, small y. And here same. Next, capital R, capital R, capital Y, small y. Capital R, small y, small y. Capital R, small r, capital Y, small y. Okay? Am I right? Next, capital R, small y, small y. Here, capital R, small r, capital Y, capital Y. Correct? And then capital R, capital Y, small y. And then here, small r, capital Y, capital Y. Here, small r, capital Y, small y. And here, capital R, small r, capital Y, small y. And then here, capital R, small r, small y. Okay. And then here, small r, capital Y, small y. And here, can you understand? So like this, you have to do it. Okay. Now, what next? That's very important. You make a table. Serial number, genotype, genotypic ratio, and then phenotypic ratio. Okay. Serial number, genotype, genotypic ratio, phenotypic ratio. What is the first one? This one. Okay, just strike it. And for understanding, I'm just numbering the squares. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Okay, what is the first one? Strike it out and write it. Okay, first one. It is capital R, capital R, capital Y, capital Y. How many were there? One, just one. Okay, here you can see the character only once. Okay, and then list the second one. What is the second one? Capital R, small capital Y, small y. This one, capital R, capital Y, small y. How many are there? Box 2 and 5. Can you see? And then list it here. 2. And then third one. Okay. What is the third one? Capital R, small r, capital R, small r, capital Y, capital Y. How many are there? Look at that. 3 and 4. 3 and 9. Can you see? 3 and 9. Same genotype. Right. So how many totally? 2. And then fourth, try it. What is the fourth one? Capital R, capital Y, small y. Okay. Capital R, capital Y, small y. Okay. Are you listening? Please listen. How many are there? 1, 2, 3, 4. All are same type only. Heterozygous condition. So how many were there? 4. And now fifth one. What is the fifth one? Look at that. 6. This one. Capital R, small y, small y. Capital R, small y, small y. How many are there? Just one. Okay. How many? Just one. And sixth one. What is the next? Capital R, all other things are small. Only one capital R, Y, Y. How many were there? Like this? 2. Look at that. 8 and 14. Yes or no? 2. And then seventh one. What is the seventh one? Small r, capital Y, capital Y. Small r, capital Y, capital Y. How many were there? Just one. Okay. So put 1. And then 8th one. What is the 8th character? Small r, capital Y, small y. Small r, capital Y, small y. How many? There were 2. Okay. 12th and 15th. What is the last one? Small r, small y, small y. How many? 1. Okay. So you listed all of these. See, listen. This is actually called what? Genotypic ratio. What is it? 1:2:2:4:1:2:1:2:1. Actually, like this only you can work out genotypic ratio. Okay. Now write the phenotype of this. What is the phenotype of this? If it is capital R, capital Y, what it will be? Round yellow. Okay. And then this one also round yellow. This one also round yellow. This one also yellow. And then this one round green. Okay, round and green. Okay, this one round and green. This one wrinkled yellow. This one and last one wrinkled green. Can you see? Then look at the ratios. How many were round yellow? 1, 2, 3, 4, 5, 6, 7, 8, 9. All these were what? Round yellow. Okay. And then round green, how many? 3. Okay. And then wrinkled green, how many? No, wrinkled green 1. Wrinkled yellow 3. Okay. So genotypic ratio is 1 is to all these things. You have to write what is the genotypic ratio equal to, how you can write? 1 is to, no, 1 is to 2 is to 2 is to 4 is to 1 is to 2 is to 1 is to 2 is to 1. This is genotypic ratio. 16 it should come. 1, 2, 3, 4, 5, 9, 10, 11, 12, 13, 14, 15, 16. Okay. And then phenotypic ratio is 9 is to 3 is to 3 is to 1. Can you understand? So this is how you should able to derive what is genotypic, phenotypic. And this 9 what? Round yellow 9, round green 3, wrinkled yellow 3, wrinkled green. If you work out this, then it looks sensible. Okay. What is the significance of this? What law can be? This everybody can work out. What is the significance of this? Grohar-Mendel derived law of, third law of independent assortment. What is this law? Law of independent assortment means, how can you write this? Inheritance of two parental characters is independent of each other. What is it? Inheritance of two parental characters are independent of each other. Can you understand this? For example, what is the parental character we selected? Round yellow we selected. Wrinkled green selected. Both are there or not? Yes, round yellow is there. You can see round yellow, one parental character. Wrinkled green, one parental character. It is there, the first two parental character. It is there or not? Yes, it is there. But also opposite. What is the opposite one? Round with green crossing or wrinkled with yellow. Can you understand? So inheritance of two parental characters are independent of each other means they can go together, they cannot go together. It is depends upon the how the gene shuffled. Okay, there need not to be. For example, Appa is tall and dark. Actually, colors and height are many genes determines. But still, for better understanding, I'm telling you, Appa is tall and dark. Amma is short and cute. Okay, the progeny can be like Appa, paiyan, like Appa, tall and dark. Ponnu, Amma madriye, short and cute. Okay, but ultava irkalam. Also, எப்படி இருக்கலாம்? பையன் அப்பா மாதிரி tall, அப்பா மாதிரி டார்க் இல்ல, அம்மா மாதிரி ஃபேர் கியூட். கேன் யூ அண்டர்ஸ்டாண்ட்? பொண்ணு அப்பா மாதிரி டால், அம்மா மாதிரி ஃபேர். இல்ல ஆப்போசிட் டு தட். பையன் அம்மா மாதிரி ஷார்ட், அப்பா மாதிரி டார்க். ஓகே, சிமிலர்லி பொண்ணு ஆல்சோ அம்மா மாதிரி ஷார்ட், அப்பா மாதிரி டார்க். சோ இந்த டால், டார்க், ஃபேர், ஷார்ட் எப்படி வேணாலும் ஷஃபிள் ஆகி வரலாம் இல்லையா? that law, that is the principle. Independent assortment. Two parental characters need not to be inherited, may be inherited, may not be inherited. That is what law of independent assortment. So they are the four important, means three important laws of genetics. First law of genetics is law of dominance. Second law of genetics is law of segregation. Both are derived from monohybrid cross. Third law is derived from dihybrid cross. So Mendelian concepts based on this only. Thank you so much for your patient listening. And tomorrow I want you people to work out monohybrid and dihybrid cross and show me. That's your simple assignment. Okay, please work out honestly. And what I have followed, all these things you have to do it. Okay, thank you. Yes, this is monohybrid cross. Hari, I gave you a task. Perfect. Mike, thanks.